It's a great pleasure to introduce now Dan Iancu from Oregon Health Science University. Dan is an assistant professor in the behavioral department of neuroscience, and he has developed some very interesting ways of adapting co-expression methods for RNA-Seq data. And I thought he should present his methods to you, because many of you deal with RNA-Seq data.
So thank you, Steve, for inviting me here. I'm very excited, and I feel like home, because we work on the same kind of concepts of integrating data and looking holistically at what goes on in a biological system. So the first, for a few years, I worked with microarray data. And then about a couple of years ago, me and my collaborators started using RNA-Seq data. And we were talking about it. And what transpired was, like, wouldn't it be a waste to just utilize RNA-Seq data the same way we use microarrays for years now? Because RNA-Seq data really gives you a lot more information, and in particular, exon-level information. So if we have exon-level information, then we can look at splicing. So let's review a little bit what gene splicing means. So if we have a gene, the kind of standard way that the gene is put together during transcription and translation is just to paste the constitutive exons together, and then you produce the protein. However, most of the genes in the human, especially in the human and mammalian transcriptomes, have variations. So an exon can be skipped, so you just produce the same gene but without that particular exon. Or you can keep an intron, and then the intron becomes part of the protein at the end. Then there are more variations on this theme. You can have mutually exclusive exons, or alternative splice sites, and this is just scratching the surface. So there are many variations on producing a protein. Okay, so now we know from Steve's work that gene expression appears to be a coordinated process, resulting in a scale-free network structure. So the questions that I want to address today are the following. Is the production of distinct isoforms also coordinated, a coordinated biological process? Also, can we describe this process using the network terminology? And once again, I don't know why this appears in this shape. But in any case, the question is, can we describe coordinated isoform production using network terminology? So as an example, let's look at this D2 receptor gene. So if you work in neuroscience, this is a dopamine receptor, and it has like an important function in striatum and even in the cortex. So it produces many isoforms, but in particular has a long and a short form. So these isoforms have different properties in the protein. So if you have the long one, that behaves differently than the short one. And that's an important biological event, and we want to describe that. So what do I mean by co-splicing? So let's suppose that we have one gene, and every once in a while, it skips an exon. Now you have the second gene, and every once in a while, it keeps the intron in the isoform, in the transcript. So my working definition of gene co-splicing is I want to evaluate whether these genes coordinate, these two separate genes coordinate their isoform production. So whenever this one keeps the skips the exon, the other one will keep the intron or variations on this theme. And of course, we have thousands of genes in the transcriptome. Each one of them will produce possibly several isoforms. So what I want to do is to integrate all of this information in the WGCNA framework.
That's a quick question.
Yes?
So fundamentally, you're suggesting that these aren't mistakes.
No, of course not.
Yeah, right?
Yes. So how do we evaluate whether they are mistakes or not? So there are different schools of thoughts on this. So some of them say that most splicing is really a mistake in the splicing machinery. And some other people say no, that isoforms really provide different functions, and they should be integrated in the analysis. So why do we want to analyze this in a framework, in a network framework? Because if there are splicing mistakes, then the assumption is, the normal assumption to me, is that they proceed independently for different genes. So if you have a lot of isoforms, but they're uncoordinated, then it's unlikely that they have any functional relevance. So that's one motivation for going to a network analysis. So when I embarked on this project, I looked around in the literature and said, is there any biological evidence that there is like coordinated splicing in the transcriptome? And it turns out that, yes, there's a fairly large body of literature that says that groups of genes coordinate the isoform production, and you can detect this in disease and normal tissue and so on. And they also think, they talk about the complex splicing code. What does that mean, and how can we describe that? So I said, okay, this sounds really interesting, but they were using these terms, and to me, they didn't exactly explain what is the splicing code. So the goal of the project, quantify the level of co-splicing using network and graph theory concepts. And in cancer, so many changes occur in a cancerous cell. But one of the most striking examples is the tumor progression. It's associated with a coordinated splicing control. So lots of people are interested in how this alternative splicing is coordinated or misregulated. So you have to compare healthy and disease tissues and see, is there changes? Are there changes? So one of the first things I did when confronted with RNA-Seq data mapped to individual exons, I looked at the correlations between individual exons and correlations between the genes. So you see here that at the gene level, these two genes have virtually no correlation. However, if you look at the exon level, you find like fairly large correlations here. So this indicated to me that exon level information is different from just kind of compressing or putting everything under the gene expression level. So by now, you're all familiar with gene expression level and how they are coordinated across different genes. Now, gene splicing, the goal is to say, okay, these two genes, we start producing isoforms, coordinated manner, and how to evaluate this coordination. And the goal is to integrate using WGCNA. So now imagine that you have your RNA-Seq data and you mapped everything to exons. So our virtual gene here has just like 3 exons. How do you represent this data? So the simplest thing is to just say, okay, I have a 3-dimensional vector, and I just record the expression level of each exon, and at the end of the day, I get this vector for one isoform, or exon inclusion profile, to be correct. Now you have the same gene, but in another sample. Once again, you just like project the vector or record for each exon the expression level, and you end up with another vector. So now, how do you compute the differences between these two vectors? So there are a lot of distance measures that are available to compare multidimensional data. So maybe the most important step is to choose appropriately the distance measure. So you can see, you can look at the angle between them. You can just look at the Euclidean distance, or you can choose yet another distance measure. So my favorite distance measure is what's called the Canberra distance measure. So the simplest one would be the Manhattan. So you just, for each exon, you go and you say, what's the absolute level difference between the expression, the number of counts for this exon and for this other exon, and you add it up for all the available exons. So this should be very similar to just comparing the absolute expression levels of the gene. The Canberra distance measure is a little bit different. So you still subtract the expression level of each exon, but you kind of normalize it, but the sum of the counts for those exons. So for instance, suppose that you didn't normalize your data using RPKM or FPKM or other available normalization procedures. This will accomplish that goal. Even though if you did normalize the data, in my experience, it doesn't make much difference. So you can use RPKMs, exon level, or you can use just counts. And in my experience, it didn't change much, the final results. And another distance measure is the cosine distance measure, which for our discussion today, we'll just imagine that it's the angle. It's related to the angle between those two vectors. So what I'm going to show you today that choosing an appropriate distance measure has important effects on the analysis. So different distance measure will capture different biological properties of your data. So here is just once more, how do you compute the Canberra distance measure? You go exon by exon. We have 3 exons. You subtract the expression level, and you normalize by the sum. So if you're interested in just kind of a differential alternative exon usage or differential splicing, there's a great tool out there. It's called Splicing Compass. And they use the cosine distance measure and combine with a kind of F-test. So they compare the distances between the samples, between the groups, with the distances within the groups. And you can find differential splicing just using distance measures. So that's an alternative to other packages out there that kind of infer the probabilistic kind of expression level of each isoform. But here, you're not dependent on the annotation. So it's not even known what kind of isoforms are present in each tissue. And you have exon level data. And from that, you're trying to infer the expression level of each transcript. And that's a tough project, in my opinion. So I mentioned that different distance measures capture different kind of biological aspects of your data. So imagine that for one of your samples, you double the expression level of the gene and of each exon. So what happens with this vector here? So all of a sudden, it becomes twice the length, right? But the angle doesn't change. Now, if you're interested in just splicing and you don't want to be contaminated by differential expression, this is the distance measure that you should use. So this will just capture alternative splicing, but it will be insensitive to just overall changes in expression level. On the other hand, if you have a situation where just this exon changes, so how do we capture this here? So just one exon changes, so then the angle will change, and so will the overall length. So the Canberra distance measure will capture both changes in splicing and in expression. Okay, so now suppose that you compute your distance between all the samples according to some gene. So our goal is to produce like an adjacency matrix, and that's based on correlations. So now we're going to correlate kind of distances. So I will give you just like the classic example of like how do you go from like, how do you generalize the concept of correlation. So imagine that you are in Europe and you find that there are similarities between the languages that people speak and also the genetic makeup. So how do you relate that? And you also measure their kind of geographical distance. So now your hypothesis is that individuals that are in geographical proximity are also closer genetically. So now how do you test this hypothesis? So we'll generalize the concept of correlation. So here we are in Europe. You measure geographical distances, and you also measure genetic differences. So you capture all that information in a square matrix. So here is the number of individuals that you sample, and you could produce a square matrix of all the pairwise differences. And now you have another distance matrix that captures the geographical distances. So what you do, you take all the unique distances which are below the diagonal, and you put them in a vector, and then you just use the Pearson correlation on this long vector. It's also possible, it should be possible to use the bicorrelation, but I haven't done that as of yet. So now let's get back to our RNA-Seq data set. So you have one gene, you measure the exon levels, you use the Canberra distance, and you produce this matrix for one gene. You repeat the process for all the genes that you included in your analysis. So now you end up with n genes. So here you have the samples, and here is how related they are according to this particular gene. Now you have n matrices, and you just correlate them pairwise. So each gene gets correlated with every other gene. And at the end of the day, you have like your adjacency matrix. So here you have genes and genes. So all the other steps are similar. So from now, you have your adjacency matrix. Everything else, it's the same, just using WGCNA. No need to reinvent the wheel, you know. Just jump on the bandwagon. Okay, so now I will go on and say that the cosine distance measure, remember, it's insensitive to situations where all exons change expression level simultaneously, but it's sensitive to changes in the ratio of relative exon usage. And once again, the Canberra distance measure is sensitive to both. So that means that we can compute like two networks. So the first one is I call the cosplicing to reflect that it's just cosplicing. So you're not going to find any information about the co-expression unless you look at one exon genes.
And why don't you use Euclidean distance here?
For simplicity. Because Canberra distance measure, I say, okay, this is, I compute the difference between two exons, and I normalize by their sum. But Euclidean, it's probably giving you very similar results. Yeah. So, yes.
In this case, what you're actually calculating is a differential network
No, it's not a differential network. A differential network would be where would be different. So in differential network analysis, you change, you do the difference between the correlations, and you cluster that. If that's my understanding, but
Distance, maybe what you calculate is the distance between A to genes from two different conditions.
the distances are between samples. So let's go back a little. Okay. So I have here my gene, and it has 3 exons.
These samples are from same class.
The samples, so the samples could be from the same class, or it could be from different classes. So the difference are between samples. So if you want to do differential exon usage, then you take these distances and do an F test. So this idea of using distance measures, it's been around in metabolomics and even in genetics. It actually started in 1967, where Mantel used it to cluster cancer data according to different measurements. So then it took a life on its own in ecological, numerical ecology. So it's kind of a workhorse of numerical ecology to compute distances between samples or, based on different characteristics. And I would highly recommend you to kind of read a little bit the literature on numerical ecology, because they have been analyzing kind of complex and dispersed data sets for decades. And they developed like really beautiful methods. And I think we should move from the concept of like kind of like ecological landscape to kind of cellular landscape. And I think utilizing distance measures is one way to get us there. So for instance, in metabolomics, they have a refinement on the Canberra distance measure. So imagine that you have the measurements for each peptide, and you want to find the differences between your samples. But you don't trust all the measurements equally. So then you kind of have a measure of how trustworthy is a particular peptide, or in our case would be an exon. So then you can weigh your Canberra distance measure and say, okay, this is how much I trust you, and this is how much I'm going to include you in my distance measure. So another application is like you have a protein, and then you measured it using like some very advanced technology, and you get like a 3-dimensional object. So now you want to compare it. So how are you going to compare like two complex objects? So the distance measures is like a well-developed methodology to kind of like get us to the point where we can compare complex data sets or complex 3-dimensional objects. So let's suppose that a few years from now, you don't work on transcriptomics anymore. You have some other very complex type of data. And you look back and say, oh, wouldn't it be nice to use kind of this network methods on this new type of data that our lab has started to utilize. So for instance, like you have here high C data, where you gather information about how like the chromosomes and the genome kind of folds on together and becomes from a, I don't know, I think it's like about 6 feet long string, becomes like a tiny object. And it doesn't become knotted. So imagine the evolutionary process that gave rise to that really complex object. But what you have is information about the conformation of this object. And people have been describing them as fractals or really complex objects that kind of have loops, and they have very interesting spatial properties. So that's your data. And you want to compare them because you notice that in disease, this object looks different. So how are you going to integrate all that data and maybe even use network methods on them? And I also believe that using this kind of distance measure approach is very useful for kind of a collaboration between computational people and experimentalists. Because, when we talk to kind of applied or bench biologists, it's very hard to explain all these concepts of like statistics and power and all this, mathematical terminology. But what if we could just like say, okay, what do you believe that the most biological significant features of your data set are? So that's the question you should ask a biologist if you're a computational person. And then, based on that, you design your distance measure. So that's a very standard step. So you can repeat this for different data sets and in different circumstances. So you don't have to reinvent the wheel all the time and choose different methods. And everything is kind of standardized except the construction of your distance measure. So back to our two good friends here. The Pearson correlation between the gene expression level is insignificant, but the mental correlation on the Canberra between sample distances is 0.76. And there are numerous examples of this gene, of genes that have this kind of behavior. So how does it look when you construct the cosplicing and you compare it with the coexpression and the other Cosplicing network, which is based on the Canberra? So you can see the red one is the Cosplicing, which is the Canberra distance measure, and the blue one is the coexpression, and the green one is the Cosplicing, which uses the cosine distance. So you can see that you have scale independence, and actually the Cosplicing has faster convergence. The connectivity, they are comparable, with surprisingly the Cosplicing appears a little bit more dense. So these are a little bit similar measurements. Centralization, the Cosplicing appear more centralized than the coexpression, and heterogeneity, once again, the Cosplicing comes up on top. So the important point here is that, yes, you do have scale-free structure in Cosplicing networks. So now, what I will do, I will compare the hubs in these networks. So I kind of shape the data to emphasize some points. So I choose the Cosplicing hubs here. They are in top 20% of connectivity in the Cosplicing network, but they are in the bottom 80% in the coexpression network. And here I have the coexpression hubs. So these are hubs that are only co-expressed and they are not co-spliced. And these ones are hubs that are only co-spliced but not co-expressed. And here actually is the Canberra distance, which captures both co-expression and co-splicing. Nevertheless, I want to emphasize that even the Cosplicing, which uses the Canberra, has different properties than the co-expression hubs. So what do we find? So we find that the gene read counts, so everything is based on variability. So as expected, if you look at the gene read counts, so this is the gene level summarization, the co-expression hubs are variable in that respect. while the cosplicing and the cosplicex have low variability, which is because I chose them so. But this is just to kind of visualize that we are based on variability, and there are different kinds of variability. A gene can vary in its expression level, or it can just vary in the ratio of isoforms that it produces, and sometimes that's undetectable if you summarize the whole gene. So exons per gene. So it turns out that the cosplicing hubs are basically very long genes with a lot of exons and a lot of annotated transcripts. Also, number of protein domains. So the proteins that are encoded by these genes have a lot of domains, so that means that they are presumably capable of interacting with many other genes. And coding sequence size, the same story. So cosplicing hubs are long genes with many exons and many annotated transcripts. And we'll go back tomorrow to this figure and show that there's interaction between cosplicing and coexpression hubs, but that's only visible if you use the Canberra distance measure. So how does the annotation look for these genes? So remember, this is data from the mouse striatum. So this is the figure, the slide that kind of sold my method to my collaborators. If you look at coexpression hubs, they're short genes, and they're involved in things that I would describe as kind of energy production. If you look at the cosplicing hubs, what you see? Neurogenesis, synaptic gene, neuron differentiation. So it was very striking. And that's because in the brain, a lot of the receptors and the synapses and even the axons, they are complicated objects, and they use really a lot of long genes with complicated structures. And I would argue that also they have a lot of isoforms. So if you look at the annotation of these genes, you will see the synaptic. And if you look at the Go and you collect all the genes that are involved in synapse, you will find that they are long and with many exons and many annotated isoforms. So the fact that they are long, it's kind of confounded or intersects with the fact that they are involved in neurophysiology. So what is known about long genes? So this is a slew of recent papers that look at the length of a gene. Does that have any particular relevance, especially in the brain, just because they're long? So autism genes, they're all long. So that's not just kind of detected using transcriptional methods, because you might argue that, yes, you use RNA-Seq data, and it's more sensitive to long genes that are highly expressed. So even if you use just association studies, so the genes that pop up in autism using just like genetic markers, they're all long. And that might be confounded by the fact that kind of genes active in the brain are long, but still. The same story here. So gene length in neurons. So there's, once again, a growing body of literature that gene length and transcriptional mechanisms are very important in neurodevelopment. So here is some speculation of why that might be the case. So how long does it take to make a transcript? If you're a long gene, it might take up to 2 days. So there's a lot going on in behavior, in animal behavior, in human behavior in 2 days. If you're a short gene, it takes minutes. So, it's very fast. There's no time for disruption from behavioral events. So there's another kind of mechanisms that it's at play here. And I'm not a molecular biologist, so I can't go into too many details. But when a cell divides, so there needs to be a copy of the DNA that's produced. And at the same time, maybe there's a transcription that needs to happen. So there are two different machineries that go along the same DNA, and one tries to copy the DNA, and one tries to transcribe the genes. What happens if they come head-to-head? That's called collision. Until recently, it was thought that neurons don't divide, but we now know that in some cases they do. But generally speaking, like neurons don't divide that much. So that's kind of one mechanism that is thought to be responsible for the bias towards long genes in neurons. They don't need to divide that much, so they can afford to have long genes. But if you have in some other organ of the body that needs to divide a lot, that might not be the case. So now let's look at the evolutionary history of the hubs. So I looked from like yeast to humans. And this is kind of a rough order of complexity. So here, unfortunately, you cannot see the top of that. But basically, I look how many homologs are there for my Coexpression hubs, for my Cosplicing hubs, and for all the other genes in the network. So it turns out that the Cosplicing hubs are more likely to be preserved or have homologs all the way back to yeast. And here I looked at unspliced transcript length. So you see, like, for the Cosplicing hubs, you see that it's a tremendous increase in intronic length. And that's not true for all the genes. I mean, most of the genes, yes, they do increase a little bit, but nothing compares to the genes that are cospliced. Number of exons, the same story. But if you look at the coding sequence length, you can go back all the way to yeast, and the homologs will pretty much have the same length. So what's going on here? So I can only speculate, but if you're now all of a sudden you use the same gene to produce multiple isoforms, like something must be able to coordinate that process. So you need to have like some control machinery. So those might be splicing factors. They are the obvious candidates here. So they need to have binding sites. So my hypothesis here is that you have like these long introns because you need splicing binding sites where the machinery can kind of bind and control all of this complicated ratio of isoform production. But of course, this is just wild speculation based on kind of computational results. Okay. So the question is that if you use different regions, yes, some of them more preserved yes, across species, yes, then would you see this? Yes. So this is a more general question about module preservation versus module differences. So it's my understanding is that you can have both module preservation and module disruption in the same data, and both will be statistically significant. If you think of an organ across species, it's both preserved and altered. So the same way with a module. So you can say, okay, it's the same module, and that's preservation, but it's also disrupted. So there are different methods to kind of evaluate this. So for module preservation, you do here what I did here. You detect your modules in the mice, and then you take the same module assignment, and I say, can I find it at a significant level in the macaque and the human data? And that's a different question from taking the same module in the humans, and one is from normal and one is from Alzheimer, and say, is this module disrupted?
Maybe we'll discuss it later.
Yeah.
But,
sure.
Do you have other slides? Or
I have just some summary slides.
Okay, maybe we'll finish it and then we'll ask questions.
Okay.
Yeah.
So basically my conclusions, WGCNA can be used to construct consplicing and cosplicing networks. And these are scale-free and comparable to co-expression networks. The distance measures are sensitive to different biological features. Co-splicing hubs are long genes, highly elongated introns, and the modules are highly preserved across brain regions and species. And some future directions, maybe doing some, that's gone. So future directions are really doing the downsampling, like it was suggested, and seeing how robust this is in different statistical terms. Yes?
Your idea of having, being able to afford having long genes and neurons is really interesting. Actually, all of this is interesting. It's stuff I've never thought about before. Would you, given that, would you expect that cells that have high turnover, such as like cells lining the intestine that turn over every 3 days, would have generally an upregulation of short genes and downregulation of long genes? I guess what I'm wondering is how many of these long genes truly are neuronally specific, or are they more related to a lack of proliferation and turnover? Does that make sense?
Yes. So the question is like, are cells that are have high turnover? What do I expect to see there, right? So the depends how many collisions do you expect. So you can have long genes, but they might not produce many isoforms, right? And you have to think about the collisions. Like, do you need to think about like, okay, I have to divide the cell, I have to produce genes. So hard to tell.
So one thing that Dave Gilbert's lab at Florida State studies DNA replication timing, and how the timing of when different segments of the genome are replicated in different cell lines, you can use that to identify cell features.
Okay.
And so I'm also wondering about that in terms of cells that undergo rapid turnover versus most neurons that are post-mitotic. And how, you know, if you're thinking about that collision of DNA replication machinery, well, maybe some of these cells are already geared that they don't have collisions because their replication is already orchestrated in synchrony with how the genes are being transcribed. So is there another reason besides that? Like, are there other hypotheses that you could think of? I know it's a really big sort of zoomed out question.
Yeah, so the question is like, what is the transcriptional machinery and the replication in different cell types, right? So I don't think that this can give an answer to that general question. So this idea with the long genes and replication came from the autism groups. So I'll direct you to their website, and they have a long discussion about collisions and why they happen more or less in neurons versus other cell types. Oh, yes?
So are a single gene, like totally out of this picture, right? It's not included. If the gene has a single exon, then it's not used at all for this analysis.
Okay, so
Single exons.
can you repeat the question?
Yeah, well, I just want to make sure that genes with a single exon is not used in this analysis at all.
You see, okay, so the question is about single exon genes. So is this included in the analysis, right?
Yes.
Okay. So yes, so the only problem with single exon genes is when you compute the cosine distance, because obviously there's no angle between one-dimensional. So over there, for the cosplicing network, I just use the Canberra if there is a single exon gene. And the Canberra, it's applicable to one-dimensional data.
Yeah, because the Canberra has this denominator. I'm just wondering, like, if you have low sequencing depth, like A divided by 1, 2 versus 1 is divided by 100 and thousands, your angle estimation for those genes could be vastly different. And just, is that an issue at all?
So one reason to use the Canberra is because we understand its properties. So if you have a 7 exon gene, the distance will be between 0 and 7. And then if you're saying that for the cosine distance now, do we understand its properties? So over there it's more tricky, because if you have a really small exon, you know, how much does that change the angle. So you have to be careful about how you use the distance measure and how you compute it. And also you have to be confident of your reliability of the measurements. So future direction is to say, okay, I believe this exon is kind of noisy and is slowly expressed, and I want to weigh it like less.
Another question I had is, like, I see that some of this module size is really pretty large, hundreds of genes.
Yes.
It's like, I just wondering how tight the cluster is. Like, you show some like raw data, exactly how the splicing patterns change between the different cell types. Like something like that.
Okay, so the size, yes. So the size of the modules, you know, I think that depends on what kind of biological question you're asking. Because I've seen people use hundreds of modules, and I've seen people use, and myself, sometimes I use fewer. So it depends on the level of resolution that you're interested in. So that's a general question about, you know, clustering and how many modules and what sizes you should get. So it's not specific to cosplicing. So I use kind of roughly the same size and number of modules in the Coexpression versus cosplicing, just for comparative purposes here.
Great. Well, thank you very much.
Thank
Very
you.
exciting.